Yes, welcome to the day 8 session on genomics and proteomics. So, in this 8th day session in Internship and Certificate course in AI and ML approaches of drug and vaccine design. Okay, so before going to today's session topics, can we recap the last session topics? So, what exactly did we discuss in the last session? Do you remember what exactly did we discuss? You have an idea now? On yesterday, what did we discuss? So either unmute and answer or you can type the same in the chat box. Yes, we started with energy minimization and MD simulation. Okay, so actually we also discussed on molecular mechanics and then we discussed three types of molecular mechanics. What are the three types of molecular mechanics or dynamics we have discussed on yesterday? Classical, very good. Number one, by classical, what else? See, the others participants can also interact. So, classical, statistical, and the final thing is the quantum. Excellent, excellent. Okay, how about the others? The SVC students are not interacting from the beginning. What happened to SVC students? You two are receiving the certificates, isn't it? Try to interact, okay? So, pride is with your hands also. Okay, now let me go with the next question. The next question is related to energy force fields. What are the types of force fields we have discussed? Types of force fields. That classical force field, charm, MMFF, all comes under the classical force field, where it uses Newton's law of motion. And if you see the next is polarizable, very good, reactive. I also need answers from the other participants. Coarse grain, superb. Good. So these are the very good PFF, polar force field related to polarizable. Good, good. So you have given that tool for polarizable. Okay, so these are the types of force fields we have discussed. The next question is, what are the types of energy minimization methods we have discussed? Types of energy minimization methods. We have discussed that after calculating the total force, our objective is to calculate the energy, and then we have to do energy minimization to make the molecule more stable. Very good. Number one is steepest descent. These are the broad classifications. We have many subtypes. Very good. Steepest descent and the conjugate gradient. Excellent. So these are the two types of energy minimization methods we have discussed. And GROMAX, I didn't take you much, I believe. I just taken you like what are the different analysis is. Can I ask you a few questions related to that? So in MD simulation using GROMAX, okay, what are the uh, uh, so in MD simulation using GROMAX, we have discussed different analysis, isn't it? MD simulation analysis. What are the different MD simulation analysis we have discussed using GROMAX? Can I give you a hint? RMSD. Like RMSD, what are the different analysis I've explained you on yesterday? SVC students, please interact. Then I'll be giving the feedback to our HOD. RMSD, RMSF, good. Like this, what else did we discuss? Radius of gyration, superb. RMSD stands for the stability of ligand binding with the protein. RMSF stands for carbon alpha fluctuations of the protein. And radius of gyration stands for the moment of inertia. Okay, that is the moment of inertia, the angular momentum. These are all measured through the radius of gyration. So between the molecules. Then what else did we discuss? Yeah, come with the answers. See, this is most important in drug design. One should know about talking. One should also know about simulation. I forgot to show you tutorials from Gromax. Today, I'll be showing you the tutorials from Gromax, and then I'll be going across with the genomics and proteomics. Hydrogen bond accept. Hydrogen bond, number of hydrogen bonds. Good. Number of hydrogen bonds. That's what you mentioned as hydrogen bond. And then what else? Did you come across PCA? Can someone expand what is PCA? When we have to go through the PC analysis, and I believe I also explained on energy. We can also calculate energy. Like however we calculate energy through docking, even the similar kind of energy we can calculate here, but we have types of energy. What are the types of energy we have discussed on yesterday in MD simulation analysis? You remember I explained about three types of energies based on ligand and receptor interaction, based on the ligand alone, receptor alone. PCA stands for your Gemini is not helping you, not myself, and the Gemini is not helping you. Oh, good, very good. The answer is principal component analysis. And how about the energy? Good try, but I expected something else. Can I show you once more? Because if one can able to interact, no problem. No one is interacting, so I can show you that. MMGBSA, LIE, you remember PBM, PBMSA. So I can show you and explain. So these are the things. MMPBSA, MMGBSA. So that is molecular mechanics generalized bond surface area, molecular mechanics Poisson Boltzmann surface area. So this is for one trajectory, this is for three trajectory, linear interaction approach for two trajectories. So binding free energies can also be determined using MD simulation. And PC analysis, principal component analysis, if we go with more carbon alpha fluctuations in RMSF, then we go with PCA analysis, principal component analysis. Very good interaction from your side. Now let me show you something with respect to the Gromax tutorial. Like I told you, we have plenty of programs. I mean, we have to use the commands for the programs to work with substeps of Gromax. But if you want to initiate with the Gromax, unfortunately, the website is not opening. The URL is mdtutorials.com/gmx. This is the tutorial website for working with MD simulation. So for downloading the Gromax tool, we have to go with gromax.org. And for working with a help tutorial, we can go with mdtutorials.com, and GMX stands for the Gromax. Here you have protein ligand complex. So this is the one we try if you have work with ligand receptor complex. So this is working actually if I click that specifically. So for this, you have to click the start the tutorial here, and then you can see the substeps. So first you have to generate the topology, then go with different box and so. Like however we come across these steps in the Autodoc, here also we can able to find the steps. Okay, these are the major steps. Inside you can find so many sub steps. So generate topology just to generate topology parameters like hydrogen atoms adding, ion adding, and solvate is for solvation to find the binding sites. Adding ions, here we are adding the ions inside, and then go with the energy minimization protocol. Energy minimization protocol is observed in molecular modeling, docking, as well as in AMD simulation. And then we go with equilibration. Equilibration, here we mean that we try to get an equilibrium between the ligand and receptor. It means that we are using NPT, the pressure, temperature, number of variables, we keep it as constant. And another thing, we keep NVT, number of atoms, volume, temperature, we keep it constant. And whatever ligand receptor complex we have, we go with the equilibrium determination. Finally, production MD. The production MD is similar to docking in Autodock. Finally, if you want to do the simulation over a period of time, for example, if you want to work with MD simulation for 200 nanoseconds, then we feed that kind of a time, etc. And then finally, we analyze there in analysis results. So it guides you step by step. As I mentioned earlier, this charm, this is for energy memorization. And if you see here, the step by step, you can find here. And yeah, initially it is asking to go with the next step like ligand topology. So in generate topology, we have ligand topology and for receptor separately. So we use this kind of a steps, and even this is a kind of an output for the step, whatever we create. So the output as well as how to make a step, everything is mentioned here. The only thing is that we should need a workstation and we need a Gromax installed in the workstation. So what command to be typed in the terminal, everything is provided, and what kind of output we get. So all this information, it guides you step by step. First try with this kind of a sample tutorial, then only we can try with our project samples. Okay, so these are something about how to work with the Chromax. Now let me go ahead with this new topic called genomics and proteomics. So if you see, these are the contents we'll be discussing it on today. HGP. HGP stands for Human Genome Project. STC, Sequence Stack Sites Content Mapping for Clone Context. Assembling the genome. You know, the genomics and proteomics is a separate subject. Some bioinformaticians study genomics 1, genomics 2 as two different subjects, and even proteomics is a separate subject. There is a lot to know about this, but I give you some basic outlines and some basic tools like how to go through these databases, etc. And we also discuss something on assembling the genome, some basic information you should know before stepping on to work with tools as well as databases. Today we discuss toxicogenomics, personalized medicine, metagenomics, comparative genomics, functional structural genomics. These are the types of genomics. NGS stands for Next Generation Sequencing. Human Proteome Project, there we can see the types of proteomics. Genome assembly. So how the genome is assembled? First of all, what is genome? How a genome looks like? So can we see it in the browser? How a human genome looks like? If you see the human genome, okay, structure. Yeah, kindly mute. Okay. So unknowingly, I think. So participants, kindly take care. Okay, with your. Okay, human genome structure. How it looks like. You know the human genome. Sarko's plot. We call this as Sarko's plot. This is how a human genome looks like. So human genome is circle in shape. Human genome is circle in shape, and if you see, it looks like similar to the DVD or CD. It looks similar to the DVD or CD for each and every genome. Okay, like if you start writing the CD, what it happens, wherever you write the information, you find that in gray color, isn't it? Similarly here also, if you see here, this is this kind of a locus maps to the chromosome 8, and this is chromosome 9, chromosome 10. So how we can define a genome? A genome is an organized information of the genes. Okay, and this is actually in circular shape. So whatever I'm telling, genes are taken from which part? So genes are taken, like for example, if I want to sequence the human genome, what is the source I can take? What tissue I can take to sequence the human genome? This is my question. You know, if we go for any heart related problems, very good. The blood is the important tissue which we use it for sequence genome sequencing. So in blood, we have so many components. What exactly we can take? I just define you. So, like if you take a drop of a blood, okay, and if you have a kind of a slide, glass slide, actually we just drop to a certain height that a drop of a blood, if it falls on the glass slide, okay, it breaks, the bubble of blood breaks and it disperses the chromosomes. So whatever chromosomes released from the blood drop, okay, this can be used for sequencing. There is something called karyotyping, which can distinguish the chromosomes. And for the sequencing, first after the karyotyping, after getting all these chromosomes, then we can go with sequencing. My next question is, how many chromosomes? We just take an example like human genome and we discuss about that, okay, from beginning. How many chromosomes are used to sequence human genome? I just showed you the size of the human genome. It is nothing but a circular shape. So whatever I'm telling circular shape, it is not a spherical or it is not a oval. So all the chromosomes, genes of the chromosome are mapped in this circular human genome. My next question is, how many chromosomes in human are used to sequence human genome? Can I give you a clue? Haploid chromosomes are used to sequence human genome. Then what is the number? 23? No, this is not correct answer. Of course, this is a national examination. In GAT also they ask this kind of a questions. 22 is also no. 46 is diploid. If you take diploid, it is 46. I told you only haploid. Haploid chromosome, total number. You are very close. Kanapriya Viramani Pragati Singh. But that is not the exact answer. 24 is right. Very good. So at last, answer from a SVC student. 24. So how we got 24? 22 autosomes plus X plus Y chromosome. Okay, 22 autosomes and sex chromosomes X and Y. So then only you will be getting totally 24 haploid chromosomes. If you see this genome, that is human genome, only the haploid chromosomes, because the duplication of genes we find in the next haploid, isn't it? So after 22, X and Y we can have. Like, however, we say this, similarly, we can also have a mitochondrial genome separately. If you want to work with a kind of a genome, I suggest you to go with the defaultly an NCBI genome. We have plenty of genome websites. For a family of plants, we also have genome, and also for a particular plant, also we have genome. So for plants, animals, insects, worms, for everything, we also have an exclusive genome websites. NCPA genome is here we find all the species genomes. So what I do is initially, instead of going like this, I better code with the NCPA, then I take it to the genome. I think now it's taking to the help information. So NCPA and then select the genome. So after selecting the genome, you can type a human or Homo sapiens, whatever keyword you know. So this will directly take you to the Homo sapiens kind of a thing. So if you see this Homo sapiens genome, what is the size? The size of the human is around 3.2 into 10 power 9 gigabase pairs. 3.2, but if you see the genomes, totally we have 3.24 million genomes deposited, which includes eukaryota, archaea, bacteria, viruses, etc. But the size of the human genome is 3.2 into 10 power 9 base pairs or 3.9 gigabase pairs. So after searching to the human genome, now only we are getting the information of the human genome. Around 1900 data are available here. And just go to the first link, which is a genome assembly link. This genome assembly can take you to this kind of information. Here you can see If you want to annotate the genes, we can find the annotation of the genes here. If you want to see the data in genome data viewer, we can click here. It directly takes you to the genome data viewer. And we also have one tool called BLAST. So this BLAST can directly take you to the related kind of genes present in the genome. We can also compare human genome with other genomes. For example, if I want to produce recombinant protein like recombinant insulin, so what genomes we have to compare? Human genome should be compared with what genomes in production of insulin protein through recombinant techniques? Can you give me the clue? If you want to produce recombinant insulin, we have to compare human genome with other species genome. What do you suggest to compare? You know which species is having more related kind of insulin? Very good. So we have to blast with the E. coli. Very good answer. I mean, generally the E. coli is used as a vector for producing the insulin, isn't it? So E. coli is bacteria is entirely different, and human is an organism that is also it is entirely different. But there is a need to know in which part of the plasmid we can incorporate the human insulin gene, so that whatever vectors we develop, we can progressively multiply, clone it, and then we can produce a huge quantity of insulin from the recombinant technique. Good, that is the correct answer. But the insulin, generally, if you want to compare with the chimpanzee, when compare, see the participants have to join earlier. So if you compare with the chimpanzee is not showing similar kind of a insulin sequence with the human. But the swine, Suscrofa, is going to show much of the similarity. So earlier in 1950s, before 1950s, where the recombinant technique was not yet developed at that time, they sacrificed numerous pigs. So they sacrificed as many number of pigs to get tons of insulin, and that's what they have taken for their diabetes. Okay, but now it's entirely changed, and this data, in some websites I can see 3.1 GB, some websites I can see 3.2 GB. So actually, if you see this kind of uh, information, is this is very bad, isn't it? The participants are instructed multiple times to accept the invitation so that it won't trouble others. Now I'm going to explain with a kind of thing. Like if you take a kind of a chromosome, how a chromosome looks like. So if it's a CA chromosome, I take a dark line and I don't find a dark line. So I take okay. So if you see a kind of a chromosome, okay, how the chromosome looks like. The chromosome is going to have a short arms as well as long arms. This is how a chromosome looks like, isn't it? In this chromosome, if you see the this part, okay, I might have taken this. If you see this part, that is centromere, as well as in the telomeres, it is very difficult to sequence. But the Illumina sequencing, next generation sequencing, can make you to even sequence few genes, but not all the genes. So only in the human genome, in some databases, they have mentioned 3.1 gigabase pairs, and some databases have 3.2. Okay, and this is the overall length of the information of the human genes. If you want to go to the specific kind of a human genome website, and especially to a particular chromosome, now here you can able to see around 22 autosomes are present and XY chromosomes are present. So 22 plus X and Y is human genome, and mitochondrial genome separately we sequence the mitochondrial chromosome. If I want to go to the particular chromosome called 11, which is of course important and related to the insulin, I'm just clicking this chromosome 11. Now we can see the in the chromosome, so many genes are mapped, and in the loci, you can able to find a particular part of the loci is also linked with so many diseases. For the treatment of diseases, obviously, okay, we check with particular chromosome. If you go with any kind of a clinician, okay, the clinician can be ENT specialist, or he can be a neuro specialist, neurosurgeon, or he can be a cardio specialist. So the doctors can be specialized in any one area, isn't it? So related to the diabetes, yeah, the doctors, the clinicians can know the information of chromosome 6 and chromosome 11. The other chromosomes are not in use. Similarly, if a doctor is specialized in neuro, then obviously for Parkinson, Alzheimer, and if he is specialized in that, he can focus on particular chromosomes alone. Okay, and if you see, so whatever chromosome that's short arm you can see at the top, and long arm you can see it here the right hand side. So a particular locus you can able to find this information. And if you see here in this, we also have tracks and user data. If you want to track by accession, so we can type the GEO or the other kind of information available. So in this tracks and user data, we have configure tracks. If I click this configure tracks, now we can able to see this information in this configured tracks, like single nucleotide polymorphism, sequences are available. If I want to focus on particular genes or products, you can see this much number of genes are present in that chromosome 11. So each and every part of the gene is linked with a disease. For example, if I go to the phenotype and disease, now we can able to see genome-wide association study of leprosy in Chinese population. Okay, and a part of a gene is responsible for lung cancer, and a part of a gene is also responsible for glioma. Whatever chromosome 11 I have taken, which is also we can find the insulin protein, code for the insulin protein, we can also find this particular locus of the gene. A part of the gene is responsible for treatment of so many diseases. Like if a person is actually having some kind of a defects, they can have a kind of a syndrome. So if only if you diagnose exactly what is the gene responsible for that kind of a syndrome or rare diseases, then it gives a way to treat the diseases through any kind of delivery, gene drug delivery, or it can be antibody related deliveries. So first we should know the sequence information. Okay, and so for this it can help you to know about this information. Okay, so genomic clones, expressions. Okay, so all this information we can find from this genome data viewer. So this is a tool to explore the data of the human genome. Okay, so we can explore in by each and every chromosome. And now let me go ahead with this kind of a presentation. So I just am explaining you about the genome. I told you how the genome looks like. It is a circular in shape, and how to search through the NCPA genome. So whatever initially we have a genomic DNA, whatever we do in our wet lab, and we get a kind of a genomic DNA from the sample, we can break into multiple fragments. We can break into multiple fragments. To break into multiple fragments, we can also use shotgun sequencing, like a shot, giving a gunshot. So that a large kind of a genomic DNA broken into multiple fragments, and this we organize it as large clone context. So this is the next step we make after blasting into multiple small genes. So initially we have to clone. Okay, we have to clone, and then if you go with this kind of a thing, we'll be getting this context, and we just try to whatever context we get, we try to sequence it to a single sequence, and we find the whatever we find the fragment. For example, if you have a fragment like a C, so here I just take a kind of a pen, because according to the example they're given like ACG. If we have ACGT as one fragment. The another fragment is CGTA. The another fragment is TAAT. So our assemble, if we assemble these kind of fragments, our sequence might be ACGTAAT. Okay, this is kind of called as a chromosome walking or chromosome mapping. Okay, so the final thing, whatever we get a final sequence, why we have to do like this? Because if you have a very large sequence, it is very difficult to sequence step by step. I mean, sequencing by each and every nucleic acid at a time. So we break into small fragments. Later, we go with the kind of sequencing through assembly. So whatever I mentioned, similarly, we can follow in quantic assembly. So whatever I mentioned, context are these small parts of the sequence. If you can uh, assemble it to a large fragment, that's what we call it as scaffold. We finally map the scaffolds. So instead of directly taking contig and mapping to a final assembled sequence, we take we use an intermediate step like merging two or three contigs to form one scaffold, and these scaffolds are further mapped to get a single sequence for the genome. So to map it, we are also using STS markers, sequence tag sites markers. So for each and every scaffold or linked with this kind of an STS markers, not only we are using STS markers to just link, this can also give some indication for a particular disease. For example, if this is linked with lung adenocarcinoma, and this is something like with a glioblastoma, actually whatever part of the scaffolds we match, okay, this STS gives you the indication of the next gene. Okay, so what is the next gene and what is it linked with the disease, etc. So we can use the STS markers to understand even phenotype of the disease. So if you see the genome sequencing, we have a high throughput genome sequencing, genome survey sequences. There are plenty of terms, thousands of terminologies are there in the genome. So we simply tell the genome sequencing can be done with Illumina, but so many technical terms are there. One should know at least the basic terms what we use. And this is actually a separate area. I am teaching something different, that is drug design, vaccine design. These are different areas in bioinformatics. Similarly, this next generation sequencing is a different area. Okay, so a lot of information we should know even in this NGS kind of genome sequencing. So assembly of context, a small fragments, if you assemble, you get scaffold, and scaffold we assemble to get the total genome. So then what is genome annotation? Genome annotation, if the genome is not just organizing the genes, the genome annotation includes to identify portions of the genome that do not code for proteins. Generally, if you see the gene, we can classify the gene as coding genes, also called as exons, isn't it? The coding genes are also called as exons, and non-coding genes is what we call it as introns. And whatever I'm telling it as a gene, etc., this is generally a nucleotide. It can be a DNA. In that, if you can able to have a sequence, and whatever we have a parts of the gene, okay, I take another color pen, and if this thing, whatever we whatever shaded part here, or the coding genes, exon 1 and exon 2, and this or intron 1, intron 2, intron 3. Okay, if only if we can able to bring the E1 and E2, E1 and E2, this is what we call it as a splicing, which we study in molecular biology by removing the introns. So we'll getting an mRNA. How we can get DNA, I mean mRNA from DNA? So just a kind of a splicing actually, okay, in molecular biology techniques. So one should know about the coding genes and non-coding genes information. that is also annotated in the genome. And we can also identify elements on the genome, a process called gene prediction. And especially if you see the annotation can be of structural annotation, functional annotation, sequence annotation, etc. In structural annotation, one should know about open reading frames and their localization, gene structure, coding regions, location of regulatory motifs. And in functional annotation, one should also know what is the function, biochemical function of the gene, what is the biological function. So sequencing a genome doesn't mean that the genome project is completed. One should also focus on the annotation of the developed genomes. Okay, so that's what you can understand from this genome annotation. And if you see this genome BLAT, this is one of the tool, like however we called as a blast tool for searching a similar kind of a sequence. So BLAT is similar kind of a tool, but it we check the similar kind of a genes one in the genome. Okay, the genome is the sequence we take for checking a homologous sequence. Generally, this can be used for even comparative genomics, to compare multiple genomes. So in the genome plat, we are seeing this kind, that is in the genome browser, UCSC genome browser, after blasting a particular gene, we can, if you want to focus this particular red mark, okay, that particular red mark is shown here. Okay, if you select a particular thing and if you blast, you'll be getting the related kind of genes information. And in genome plat, we can also see the protein-protein interaction information. The PPA interaction is also visible. So I initially searched for a gene called breast cancer antigen 1. So what is the interacting proteins of BRCA1 of human genome? So immediately I got these are the top 25 interacting proteins. So this information is also available in Genome Blast, and the genes information is mentioned in below. So if you click this hyperlink, you will be getting more information of the particular gene. So like this, you can search for any particular genes in the human genome through Genome Blast. Flowchart of genome assembly. So I already discussed now. So we have a context, and our context we map it to the scaffolds, and the scaffolds are mixed through STS to form the entire genome. So initially, you can map it to a chromosome, and if you map all the chromosome genes, you get an entire genome. So genome can be up to a chromosome assembly, or even it can be up to the whole genome assembly. And if you see in the genome assembly, I told you many technical terms you can observe. The most important technical terms are high quality reads. High quality reads. The other is low quality reads. Actually, instead of taking a single sequence, we take a clone of multiple sequences, and after going with the shotgun sequencing and making into multiple fragments, we try to go with the Illumina sequencing. For doing Illumina sequencing, an entire genome, it takes around 7 to 14 days based on the server capacity, etc. Okay, so it takes around 7 to 14 days to sequence the entire genome. Where exactly we can find the genome? Like if a patient is suffering from a rare disease, definitely the the clinician instruct to sequence the entire genome. Like however, the cardiac patients come with the ECG, EEG, etc. Now, sequencing the genome is of course a mandatory criteria to find what is the defective gene. Can be also a syndrome that definitely the doctor asks you to bring a kind of a genome. So nowadays, you can also heard something called Genome Patrika. I don't know how many heard about this Genome Patrika. Anyone heard about this? Like in the developing countries, I don't know this. I mean, we, we were not concerning about that. But in developed countries like US, they ask you like this. Like instead of showing Patrika horoscopes for matching the brides or bridegrooms, nowadays they're asking what defects. If you submit our genome, they can find the defects. They can find the defects like in 40 age they will be getting bald, and some in 30 age they will be getting bald. Even if they see such a kind of a criteria, even that kind of up to the baldness character, we can able to find it from the genome. and any kind of metabolic disorders when it can come, everything we can able to find it from this genome itself, okay, from the hereditary information. So this also linked with the genetic counseling. Okay, so nowadays there this kind of a thing is also getting popular in India. Okay, so we can find the genome is of course an essential task, and so many uh, NGS scientists are required at present for different hospitals to understand the defect gene for particular diseases. So strategies, we use the Brunges graph. We use Brunges graph for sequencing the genomes. So generally, instead of going with the protein sequencing to identify the type of protein, it is better to go with the RNA sequencing, mRNA sequencing to identify the species. To identify the species, generally we go with this mRNA kind of a sequencing. Okay, so that is if you want to find a patient suffering from cancer, what is the stage, and how and whether he can be treated or not, and what is the defective gene if they want to find. Obviously, we can go with this RNA kind of a sequencing. First, initially, we can compare with the healthy tissue. Whatever tumor tissue we take, we compare with the healthy tissue, and we can come up with a kind of a identification of the gene, what is responsible. Like generally, if you go with a precision-based medicine, whatever drugs available, whatever antibodies available in the market, we can't able to use it for all the patients. For the diseases like cancer, definitely the patient has to be diagnosed with what type of genes are responsible. Then only the treatment has to be given accordingly. For this, of course, the RNA sequencing type can help us to come up with a precision-based medicine. So we can use a gene-related targeted delivery for the treatment. The next thing is toxicogenomics. What is toxicogenomics? So actually, if you see, there are types of omics, like genomics, which study about their genes, organized collection of genes in the chromosome. And proteomics is something called the collection of proteins from an entire set of an organism. Entire set of proteins of a particular organism is what we call it as a proteome. And study about the proteome is proteomics. So generally in proteomics, we study PPI interactions. We can study peptide mass fingerprinting if you're working with a peptide-based analysis. Okay, so these are generally we study in proteomics. But when compared to the proteomics, metabolomics, lipidomics, the name itself metabolomics, what you can understand, study about the metabolites. There are primary metabolites, secondary metabolites. So whatever metabolites secreted from our body, okay. So if we try to work on that, because even metabolites can depend on the metabolic diseases, etc. So one should also can analyze the metabolomics. So generally, if you want to precisely treat a patient, okay, not just the genomics can help us. Other than genomics, we should also work with proteomics, which we can study in the form of PPA, etc. And then we can come up with metabolomics. Okay, so we have to use this as a sequence-based strategy for the treatment. Not just working with genomics or not just testing with the proteomics can give you a precise treatment for the rare diseases. Lipidomics, we have a collection of lipids, whatever the lipids are created. So if you study on the lipids, transcriptomics about RNA, this is most important. So to identify the species and the target gene or which is responsible, we can go with this RNA kind of omics, that is transcriptomics. Why I'm telling you this is important, when compared to genomics, proteomics, metabolomics, this transcriptomics is cheaper one for identifying the gene of the particular disease. So many of them go with the 16 years RNA sequencing technologies. Okay, so if you see these kind of types of omics, okay, And if you're focusing on toxicity, so one drug can be toxic to another organ. So that's most important. We should also analyze the toxicogenomics. Okay. So whatever we studied with Protox3, we remember. So in the previous session, I showed you a server called Protox3, which can determine the various organ toxicity and various pathways. You can check and you can check what other kind of a toxicity you can get. You can also get LD50 value of a particular drug, whatever we provide. So if we focus on determining the toxicity and we apply this toxicity in genomics, we call this as toxicogenomics. For working on toxicogenomics, of course, we use the same website, Protox 3.0. Personalized medicine. What is personalized medicine? Just now I told you to bring a kind of a personalized medicine for treatment of diseases like cancer. We should uh, first segregate the patients. If you see the breast cancer we have the breast cancer is majorly caused because of HER, progesterone receptors, and EGFR receptor like this. So whatever we have these receptors, other than these receptors, we also have triple negative breast cancer. Triple negative breast cancer, where if you find the absence of three important receptors which causes breast cancer, then obviously you can call it as triple negative breast cancer. So we have a different set of patients for that, the set of people. And that people can also diagnose with more than 10 different genes. Each and every patient is majorly affecting because of one particular gene. So if 10 genes are responsible for triple negative breast cancer in combination of genes, then obviously one should first initially go with all the types of omics studies. Go with genomics, proteomics, okay, interactomics. This can give you a kind of a best knowledge, not only up to the transcription data, also translational data. So this information can also give the good information. So if you want to tailor the medicine or come up with a kind of a personalized medicine, okay, so that's what we call it as a precision medicine. We can able to cure almost all the types of patients, but that should not be in the chronic stage. If it is up to 3 stages, we can cure. If it is fourth and fifth, there is no option for most types of cancer. We have to go for transplantation of the particular organ if it is feasible. Okay. So if you don't want to miss any chances, we have to diagnose in the early stage itself. Metagenomic study. What is metagenomics? Metagenomics is the study of all genetic material from all organisms in a particular environment. If we take our gut, we have so many bacteria is living, and this bacteria is responsible for the development of new diseases also. Based on the gut bacteria, we can find whether we are healthy or not. So like this, even we can find the bacteria also present in the environment. Okay, if you work on these kind of genomics, especially with respect to environment or with respect to the gut, this type of genomics exclusively call it as metagenomics. Okay, nowadays because of the gut is what we are maintaining our good health. If our gut is good, we can also live younger for many years. The bacteria present in our gut is most important. Uh, okay, now related to this metagenomics, I told you. So we study the interaction, the relation of bacteria with the human, etc. Isn't it? So that's what in metagenomics. We also study with respect to the environment. That also I've given. So that is if you study what are the particular soil bacteria, how about the seawater, human gut. So these are all considered as an environmental factors. So study about the specific examples is what mentioned is what we call it as metagenomics. Then what is comparative genomics? Comparative genomics is comparing more than one genome, like comparing Acinna pestis kim, Acinna pestis antiqua, different species strains are available. Acinna pestis is actually one species, but in that one species we have different strains. For different strains also we can compare. Especially we can try with the SARS-CoV-2. In SARS-CoV-2 we have 100 mutations and 100 strains are available approximately. If you want to compare alpha, beta, gamma, delta, all the kind of strains of SARS-CoV-2, we can compare and we can get a kind of an information. From this information, we can design the vaccine. The Covishield, whatever we take, can cure the first strain. It can't able to cure the second strain or third strain. The efficacy will be reduced. Then what is the exact uh, gene responsible? And then how about the vaccine? What kind of vaccine we can develop for the particular gene? So this we can able to make it only by comparing these comparative genomes. So we have tools to compare multiple genomes. So even beginning we have discussed, like if you want to bring a recombinant insulin, obviously we have to compare human with the E. coli. We can't compare with the pig or chimpanzee because we are using the recombinant developed technology for producing insulin. So according to the NCBI data, the estimated size of the human genome is 3.1 billion. And of course, we take 22 plus X plus Y, 24 chromosomes. And see, this is actually not updated. At present, you can see around 35,000 to 40,000 genes are available. Okay, so these are the total genes present in the Homo sapiens. The increase from 25,000 to 35,000 is just because of developed Illumina technologies and modified Illumina technologies, even the part of the centromere and part of the telomeres can also be sequenced. And so the range of sequenced genes at present is increased from 25,000 to 35,000. Like, however, you find the human genome, if you compare with the other genomes like mice, which is most widely used in vivo experiments for working with human diseases, if you see the mice, we can see around 2.9 billion base pairs or is the size. Okay, so we are also working the diseases, diagnosis of diseases of human compared with the Drosophila. I told you, in drug elucidation, we can try with the nematodes, we can Cenorbitata elegans, and we can also compare with the fruit flies. We can induce the human diseases in this small fruit fly, and we can able to come up with a kind of a drugs for even for humans, used as a model species. And for this, it's very small size, 165 million. So if you want to try like this, we have to compare the size, chromosome, etc. These are some of the basic information. Functional genomics. If you're working on a function of a particular protein, for example, if you're working on insulin, the insulin ligand is just a 110 amino acid, and the receptor is around 700 something. And if you see this insulin ligand, okay, well, not the entire protein is responsible, not the 1 to 110 amino acid is responsible for that insulin function, that is controlling the blood glucose. Only a part of the gene, only a part of the motif of the insulin ligand is responsible. I believe nearly some 97 to some 108 amino acid. Okay, this is the important motif which is going to regulate the blood glucose level. Then, if you're focusing on a particular part of a motif, and if you're working on, and if related to the gene, if this is the important gene location, then obviously one can compare with the different genomes also with respect to the function. Okay, the function is here in this case is regulating the blood glucose. So for functional genomics focuses on the dynamic aspects such as gene transcription, translation, regulation of gene expression, and protein-protein interactions. These are other examples. These are other gene ontology examples which we can use it for functional genomics. Structural genomics. If structure is most important, for some proteins, instead of function, we majorly bother on structure, especially myosin, muscle protein like myoglobin. So that we have to know the information of the structure. We have to compare the structures. NGS is something to sequence the entire genome. We can go with this NGS. Either we want to sequence the entire genome, or sequence the chromosome. So we can use the NGS techniques. The type of NGS techniques what we using is sequencing based synthesis. Sequencing based synthesis, the best example is the Illumina and pyrosequencing. And these are some of the methods. Human Proteome Project. What is Human Proteome Project? So Human Proteome Project is a collaborative effort coordinated by the Human Proteome Organization. So majorly we study a large-scale study of proteins of a specific organism. Human proteome, we study the proteins of a human. Okay. So in Proteome project, we use a kind of a workflow to link with various tools and databases. Okay. So to check for the proteome, we can go with UniProt. There is a website called UniProt. Okay. So we can go to the UniProt. In this UniProt, we can have a database called Proteomes. Here, we can search for a specific species. I search for human proteome. So in human proteome, this is the organism ID. 83,607 entries of proteins are available for this human. So other than human, we also have HPV and other kind of virus, bacteria, which is associated with human. But for the human, we have this much entries. So if you go to these entries, we get this information of a particular human proteins. So it is a collection of set of proteins of a particular species. That's what we call it as a proteome. And we also discussed something about KEGG, isn't it? So KEGG, KEGG, Kyoto Encyclopedia of Genes and Genomes. The name itself implies genes and genomes. Okay, we also have an exclusive database called Genome. In the KEGG, the genome.jp, we also have a genome database separately. If you want to find a genome information, like our research for NCBI genome, similarly here also is check for KEGG genome. Here you can type the specific species name and you can search for it. Okay, so this is about another database for genome, KEGG genome. And there is another kind of a database for proteome, PRIDE, Proteomics Identification Database. So it is present in ebi.ac.uk. Earlier, whatever I showed is from UniProt, UniProt Proteome. In EBI, European Bioinformatics Institute, this is majorly taken care by Germany. The earlier UniProt, whatever I showed, is taken care by UK, London. Okay, so in EBI, we have this kind of a database, and if you see the protein-protein interactions and proteins information of a particular species are available here. Only thing is to type the name of the protein and we have to search. There is also one more important database for proteome that we call StringDB. Remember the StringDB? Tomorrow we'll be seeing extensive information about pathway databases. There we also discuss on the string. The string DB, if you see the overall information, okay, you can able to see the protein-protein interactions. I believe I showed you earlier, like if you type any particular protein name and if you click for search, it shows you different species, what species exactly we want to go through. If I want to go with this Homo sapiens protein called TRAP1, which is also called as each of protein 75. So we are getting this. Okay, so simply you have to click continue. So whatever data that is HSP, the TRAP1 is connected with the 10 important proteins. So how we get this data? This data we are getting from this protein-protein interaction experiments. So these are the experiments performed to produce that kind of a graphical kind of an output. So what are the wet lab experiments they have carried out? Now I am going to show you. So whatever we are seeing, a kind of a graphs, etc., or a product of wet lab experiments. For protein-protein interaction, we have different kind of a data sets. The first data set, they established by anti-tag co-immunoprecipitation, where they have 1557 plus 10 different proteins. So in this technique, they established the relation of TRAP1 with around 1565 proteins. And in another experiment by another scientist, they did with anti-tag same kind of experiment, but they came up with one protein less. Similarly, you can see they come up with a different kind of a techniques. Pull down assay. According to pull down assay, they had a link of TRAP1 with nearly 118 proteins. So all these things are a proteome databases, gives something information on proteins, protein-protein interactions. Okay. So this is the major thing you can find in genome and proteome. So today we have discussed what is genome, what is proteome, because there is much information about in interactome, metabolome. So we have discussed the most important things on today, and tomorrow we'll be discussing on pathways and how to use cytoscape, and we, if feasible, we discuss on how to work on pharmacokinetics using MATLAB. Okay, so these are the topics we'll be discussing it on tomorrow. So today we have discussed the basic things. Any doubts you can ask me at present. Any queries? If no queries, thank you very much for your patience and interaction. Okay. Have a great day to you all. Bye.